Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we jump into this new episode, we have exciting news to share with you. The new Dr. GPCR ecosystem platform is almost ready for launch. What is it about, you ask? Well, think about a place where GPCR scientists, trainees, and GPCR organizations can thrive and where it's all about science and GPCRs. Access is restricted to members of the field, and each sign-up will be approved accordingly. Once your sign-up is approved, you'll be able to enter the ecosystem and interact with GPCR colleagues like never before. You can still sign up to be an ecosystem beta tester. We are going to open the ecosystem slowly to ensure that you get the best experience. Please keep in mind that the Dr. GPCR ecosystem is a unique place, a first and one of a kind, where we want to make sure that you get the best experience out of this new world. Visit drgpcr.com/ecosystem to start your GPCR journey. Also, make sure that you mark your calendar for the third edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned as we are working on the program for the summit. Visit drgpcr.com to find out about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. And today I have the pleasure of having with me Dr. Daniel Wacker. Hi, Daniel. How are you?
Yamina, good to be here. Doing
Good
well.
to
How about
have you.
yourself?
I'm good, other than the non-COVID bugs going around.
Was glad to
lately.
hear it's non-COVID. Yes.
Absolutely. I'm so glad that we got to do this. Thank you so much for coming along.
Of course. Glad
So
to hear.
let's start at the beginning. Would you please tell us who you are as a scientist and how did you become a scientist?
I'm currently an assistant professor in the Department of Pharmacological Sciences and in the Department of Neuroscience at the Icahn School of Medicine at Mount Sinai in New York City. And I've been here for a number of years now. I'm still sort of kind of in the process of establishing my own lab since, COVID has done a number, I think, on all of our careers, but I think in the midst of starting a lab that has set us back quite a bit. Obviously, I've been around the GPCR field for quite some time now, and it really depends on how far you want me to go back. But just to briefly summarize, I think the most important recent steps, that was my Ph.D. At the Scripps Institute in La Jolla, California, working for Ray Stevens, and then more recently a postdoctoral tenure at the University of North Carolina with Bryan Roth. Two big names in the GPCR field that I think don't need much of an introduction. Yeah, and that was basically the past 8 years prior to coming here of my career. So that's roughly been my track record in the GPCR field.
And as a young teenager, were you always interested in science, or what were you interested in?
Yes, I was. I think what holds true for most of us is that we're all somewhat, curious beings. I think even outside of science, that can be said. Certainly, as kids, you're exploring things, and then eventually, some of us get to be lucky enough to turn this into our careers. I feel privileged for sure. That being said, I think what also had a big influence is that my mother is a technician. She doesn't necessarily do similar type of research that I'm doing. She's not in the field of biology, but she does a lot of physical chemistry, working on a number of things. And so very early on, I had some limited exposure to scientific inquiry. Let me put it that way. It was more working for sort of a business, and they were analyzing chemical samples, and so that was how I was first introduced I'd say at the age of 10, when I want to say my consciousness somewhat kicked in, certainly when it comes to scientific things. And yeah, I mean, I spent some summers during my teenage years at that institution, and I was working there. So after that, I'm born and raised in Germany, so that was in the southern part of Germany. And I think during that time, I think that was kind of sort of the formative years that I started to explore that there's people out there that basically pursue curiosity as as a profession, and that kind of opened my eyes to the whole field of research and science altogether. And that kind of laid the groundwork. Then certainly in high school, I mean, I had fairly good teachers in chemistry and in biology. In physics, I actually was always more on the physics and math side of things than on the biology side of things, which came later. But high school then kind of sort of, laid the groundwork for me to start my college degree, which in Germany is heavily specialized. It doesn't really compare with college degrees in the US. You have to pick right from the get-go what you want to do. And so my 4 majors were inorganic chemistry, biochemistry, physical chemistry, and organic chemistry. And then later I specialized during my master's in biochemistry and structural biology. And the rest is kind of sort of history. So yeah, that's kind of been the early years for me.
What, from Germany to the US, what led you to Ray Stevens's lab?
Well, palm trees. That's probably the one-word answer. But to backtrack a little, I started exploring going overseas, and also as a young guy trying to figure out like different cultures and traveling abroad. And I actually started in Cambridge, England. I worked for Kiyoshi Nagai, which was one of the first grad students of Max Perutz, and I'm arguably the father of X-ray crystallography, or one of them certainly. And so I met Aaron Klug, I met Venki Ramakrishnan, I met some of the big names in the field, and that kind of formed my opinion on structural biology early on. And I returned to Munich, where I actually did my college degree, or my university degree, my bachelor's and my master's. And I during the later years worked for Patrick Cramer and Roland Beckmann, both working on, I'm going to call it the biochemical dogma. Roland Beckmann famous in the field of ribosomes, Patrick Cramer famous in the field of, now also mediator complexes, but RNA polymerase too. And so both heavily structural biology, like both working heavily in structural biology. I thought the technology was very interesting, but I feel like the biology wasn't really what I was interested in. It sounded interesting to translate DNA into RNA and go into proteins and all that, but it was too fundamental for me. I always felt like I wanted to have something, and I think we all lie to ourselves what that really means, but I always wanted to have something that's more translational, which is why later it led me to GPCRs. But from these labs, I actually did my master's project, not the degree, it was still from the University of Munich, at the Rockefeller University. And I worked for Guenter Blobel, who's since passed away. Another big name, the Nobel Prize for signal and sorting of proteins. And there I met a number of people, and I started reading more and more papers, and it became increasingly clear to me what I wanted to work on. And with the help of a colleague there who'd done his PhD at Scripps. Scripps was high on my list of one of the institutions that I wanted to, or I considered doing my PhD at. And at the end, it came down to doing my PhD either at Scripps or doing it in Switzerland at the University of Zurich and ETH. They were a combined program. And because both interviews were within a month in February, March, to circle back to that, the deciding factor were the palm trees, because once you step out the plane in Switzerland, there's no palm trees there, but you step out the plane in San Diego, and it is wonderful and warm. They knew how to recruit people. They definitely did. And then meeting Ray thereafter, I had signed up, and he was a great mentor, I thought, and yeah, things took off from there.
That's so funny. You mentioned, stepping off the plane in February in California, and I feel you. I was at a Gordon Research Conference. I think it was in January. It was a molecular pharmacology, almost 10 years back, and I was in Montreal, minus 40 Celsius. Mm-hmm. Our plane was delayed because, right? Our plane was delayed because of all the ice on the plane wings. So we waited 6 hours in Montreal. Took the plane, got off the plane at LAX, and I mean the reaction of, we were 4 or 5 people, and we looked at each other and we're like, okay, jackets away, sunglasses out, and it just, I feel the palm trees. I feel you on that event.
Still remember that flight, flying into LAX actually from New York, and then taking the commuter plane from LAX down to San Diego, which is like 20 minutes of a flight, and you just see the coastline. And I remember leaving Switzerland, and I was like, this is where I'm going to do my PhD. And then I got off the plane in San Diego, and I'm like, oh, hold up, let's change plans.
It was a 2-for-1, definitely there. I didn't know that you were at Rockefeller University.
Yeah, that was 2008, 2009 for about a year and a half. I met a lot of great researchers there, and I'm glad to be back in New York. So it's definitely the starting point of my American career, so to speak.
I love Rockefeller. Well, as you know, I spent 5 years at Rockefeller as well as a postdoc, a little bit after you, but our main lab was on the same floor as Dr. Blobel's lab, and we collaborated a lot. And maybe I shouldn't say this on the podcast, but I had a dog back in the day. His name is Apollo, and I used to take the dog to the lab, especially in the evening if I needed to do something. And I actually bumped into Dr. Blobel, and he was mentioning that he had 2 dogs.
He had 3 when I started.
3?
So it was 3 dogs, and it was the most loyal dogs I'd ever seen. I still remember he was walking down the hallway to go to the bathroom, and the dogs were following him. He went into the bathroom, he closed the door, and the dogs were just sitting outside, staring at the door, just waiting for Guenter to come back out and then mosey on back to his office. It was a sight to be seen, that's for sure. So Rockefeller at the time, anyways, they didn't mind the dogs as much, or maybe it was just Guenter's floor. I don't know.
No, I think a lot of people have dogs. For example, Charlie Rice won the Nobel Prize. Also has two dogs, and I distinctly remember that they went to the lab with him in the morning and home in the evening, and they took them sheep herding during the weekend. So I think Rockefeller is a special place, a concentrated place for science, amazing people, and amazing dogs as well.
Yeah, for sure. Tom's still there, right? So
yes. Exactly. Tom's lab is actually on the same floor, on the 5th floor, but I was in the other building, in the Bronx building. We had to expand. All right. So GPCRs. When did you first hear about GPCRs? When did you first think, oh, actually, this is a good system to study, looking at structures and at crystallography?
I think the first real exposure I had were from some papers back in my college days. I still remember when I need to apologize to the entire rhodopsin community, but when the first beta 2 adrenergic structure came out, it was just a huge splash. And I remember one of the senior postdocs coming in and basically just like yelling and screaming at people. They need to all huddle up and they need to see this paper, and it's just absolutely amazing. So there was Brian Kabilka's work together with Ray Stevens, and basically, the claim to fame of this paper was, of course, like Brian's technology that he established to fuse T4-lysozyme into beta 2, which made this happen. And like all the protein engineers in the lab were just like mesmerized by how cool this is and it works and everything. So that was my first conscious, like experience with it. And it must have been back in 2007, 2008, right around the time when this came out. And that's when I kind of started reading a lot more about the different fields. Membrane protein crystallography was always something that I was really interested in. There was one postdoc in Roland Beckmann's lab that I worked in at the time who was really who came from a membrane protein crystallography lab, and he would always talk about how they're extremely challenging, but that's where all the drug targets are. I mean that holds to be true now. Years later when I started reading the drug discovery papers and all that, about 70% of all drugs actually target ion channels and, transporters and GPCRs, of course. And so that was when I first got really interested in that. And then that big structure had just come out, and I looked at obviously Brian Kabilka's lab, and I looked at Ray Stevens' lab at Stanford. That I don't think it was on the list for me, and it just didn't feel like something that was attainable. for me. So when I got into Scripps, which wasn't just for Ray's lab, there were other labs that I was looking at as well. I decided, to give this a shot, and I had some really good email conversations with Ray. And at the time, it was just the beta-2 adrenergic receptor and the A2A adenosine and the rhodopsin structure, of course, that came out years before those. And my interest obviously was on something really drug related. And beta-2 was interesting from the point of view of beta blockers, but certainly there were other things that I thought would be more aligned with what I was interested in. And so the first thing that came to mind, and I've showed Ray my email because he didn't remember, like at the time of my graduation, was that I was really interested in serotonin receptors. And at the time, I didn't know that about 3 or 4 years later, I would publish the first serotonin receptor structure. And so that was quite a happy coincidence, but I still have emails before I even started my PhD, exchanging notes with Ray, talking about how if he would be interested in working on something else, if I could pick my own things. And that's eventually what happened, because he was always open about these things, that he would always, like foster people that were interested in their passions and spend a lot of time on that. So that's kind of sort of the trajectory that I took there.
It's so interesting. You mentioned that, it was important for you to be able to pick a lab where, well, other than the palm trees, obviously, where you could be working on a project that is really interesting to you, that you could pick the project that you wanted to work on. And as you know, some of our listeners, or a big part of our listeners, are PhD students, grad students, potentially postdocs, who sometimes, I feel like we can always talk about, okay, how do you approach picking a lab? And how do you, and learn from other people's experiences? So you mentioned that you visited multiple labs at the Scripps. How did you, first contact Ray, and why did you pick Ray's lab for your PhD?
Ray's lab was clearly my absolute top choice. Why I wanted to come there. It was GPCR driven. I wanted to work on GPCRs. There were some other labs at the time that I was interested in, but they were more like fallback options in case it wouldn't pan out. Which, yes, aside from the palm trees, one big attraction of the American system is the rotation system, which you don't have in Europe, right? You sign up with a PI. Now there's more and more programs, but at the time there weren't that many programs, certainly not like in Germany, where I only briefly looked. And so for me, it was very important. If I sign up for PI and I don't like the work, you can never sniff anything out during a rotation. But if I don't like the work here, I don't like the people, there's something that just bothers me. And it's important to be comfortable in your PhD, because there's going to be hard times. If you already have a bad feeling walking in, that's not a good setup. And so it was important to me to have options and to be like, what, this is great, but it's not the end all be all. And well, kind of sort of turned out to be the end all be all for me. So I was very lucky that way. But that was a major point, that I would have different options if Ray's lab wouldn't have panned out. The second thing was, you need to find a mentor that works for you. I have students now in my lab. I've mentored many people over the years, and some people, they just, they need closer mentorship. They need some people even request micromanagement, and some people don't want it at all. You get a spectrum, and so it needs to be a fit. And I have colleagues here that always tell me that like rotations are like dating. You have this great option of just figuring things out on the short term, and you'll never learn everything, but you get a pretty good sense of the lab, and you'd be surprised how many people, when you press them, if they like it here, are, easily willing to tell you the full truth over a year or two and tell you, this is not the place where you should be because of ABC. So if I were to give any advice, you need to find a mentor that you like. You need to look for backup options, because nothing ever works out exactly the way you want it. But other than that, it worked out for me. So those are, I think, the 2 big points that I did consider. But we're all biased in our own experiences. We always think the way things worked out for us, that's the right way. I really can't speak for anyone else there. So.
But I think learning about other people's experiences is also a great way to figure out what you want and what you don't want. Because I think you made two great points. What I would like to add to it potentially is the fact that, first of all, you as a person have to figure out what is it that you want. As you mentioned, some people like to be micromanaged. They ask for it, others don't. But there's a third category of people who don't know what they want. They don't even know what they need. And as a person, you need to figure out what works for you, and then go look for that lab, look for that PI, but also have your options there.
Absolutely, yeah. That was absolutely right. It just felt like when I came into the lab, there was just I came in when the field exploded, right? I came in when there were like 2 GPCR structures, and then I left the lab when there were like 40, something like that. I mean, it was like an insane time. So and that wasn't me. That wasn't me like being smart or the best at what I was doing. I was just really lucky being there. So there's this element is just hugely important, and you can't control it. So it's I don't want to spread anxiety, but you need to be comfortable in what you're doing. You need to find the right environment for you, and then everything else is to some extent not up to you. But yeah, go ahead.
No, I was just gonna say I really like the comment that you made about rotations, which don't exist in Canada, at least not in my time. Yeah. You picked a lab and that was it. And the comment that you made about dating, and a rotation is like dating, and I strongly believe that it is. And even if it's a rotation or it's an interview for a job, anything, it's like dating. If the two parties or the two groups don't get along, just turn around and move on, because during your PhD, especially on your postdoc, you spend more time in the lab with your lab mates and with your PI than with your own family. It was just ridiculous, the amount of time that you spend in the lab.
Yeah, absolutely. So,
I love the dating analogy, and it's true. I totally agree.
I can't take any credit for that. I don't know if you already had Lakshmi Devi on your podcast. She's a GPCR expert, deorphanizing peptide receptors and so forth. So she's my neighbor here on the floor, and like that's her mantra, and I think she's absolutely right. And she hit it on the head there, so a little bit. And since then, I've adopted that. So I need to give her in the scope of science. I need to cite her work there. So yeah, that's
great. I'd love to have her on the podcast. Of course, I've heard of her. I know like some of her work. So I think that's going to be a great in for my email, saying I love your approach to figuring out if somebody's a fit or not, in the comparison to dating. All right. So you worked on the serotonin receptors in Ray Stevens's lab, and then you decided, okay, I have my PhD. What next? What made you do a postdoc?
Well, the postdoc these days isn't so much of a choice anymore, right? For depending on what you want to do after, unless you want to do consulting, I think it's becoming increasingly difficult to leave your PhD and even get an industry job. People do it, and some people are very successful that way. But if people plan to become big shots at Pfizer or at Merck, I mean, you have to have some serious publications and serious experience under your belt, because those aren't jobs that are just like there for pickup. So a postdoc was kind of something that I always felt like was a necessity. It was also something where I felt like, okay, so for once, I'm not sidetracked by writing a thesis. I'm not sidetracked by classwork. And I mean, there's just so much stuff in a PhD that's not actual research that, I just felt like I had more to give productivity-wise. And I do feel like that my postdoc, and I've heard that from many people, is the most productive phase that you'll ever have as a scientist. I mean, even as a PI, having only been a PI for a short amount of time, I feel like I'm nowhere near as productive as I was as a postdoc, because you can be single-minded, really hardcore on a single project, and you can just hammer away at it, and that's just the way to make progress in science. It's a marathon, and it depends on the time and effort that you put into it. Now, that being said, it was at the time relatively clear to me that I wanted to give the academic track a chance, and I wanted become a PI and see what that is like. And for that, in particular, a postdoc is just an absolute must. I mean, there's colleagues of mine who've managed to be quicker. Others are slower, but it's very, very rare. You find those people, but it's very rare you find people that transition directly from their PhDs into an independent faculty career, even though there's some grant mechanisms in place for that. And when I picked my lab, and that was Bryan Roth's lab, it was important to me to not just do the same thing over again, even though my papers might look that way. So one thing that I'd learned in Ray's lab was the structural side of things, because I'd always been a structural biologist. I received training way before Ray's lab in X-ray crystallography from crystals and so forth. So for me at the time, it was more about mastering protein purification, mastering taking pictures of things. What I didn't have down was the biology, the pharmacology behind things, and I thought that Bryan Roth's lab would be the perfect marriage for that. And I think it was very mutually beneficial during my phase, because I brought the entire X-ray crystallography operation from Ray's lab to Bryan's lab, who hadn't really published any structures other than being a collaborator with structural biologists. And so I set this up with colleagues of mine in Bryan's lab as the resident structural biologist. And in return, I learned from him and from the people in the lab more and more about molecular pharmacology and more the classics of, how to use, operational models, how to interpret what's now known as bias, but quantitative pharmacology, and how these receptors actually fit into the big scheme of pathophysiology. So it was very important to me to not just be a structural biologist in one lab and then move to another lab where I would just do more structural biology with other structural biologists. So it was more like I bring something and I get something back. And again, coming back to the fact that I only know my own story and it's worked out so far, that's something I always advise people. It's what I've advised my people. Don't just copy paste. If you've done your PhD in a cryo-EM lab, just going to another cryo-EM lab that works on something slightly different. I mean, it can lead to a good career, and that's not what I'm saying, but you might be bored out of your mind doing the same thing over and over again. You certainly won't grow as much as a scientist just because you just need a comprehensive look at things.
I love the advice that you just gave, is really looking at something, and also the fact that you joined Bryan Roth's lab. You brought something, you established something in the lab, but you also learned something that you took away from your time.
I learned a lot. Yeah, I can't thank him in the lab enough for teaching me. I mean, again, I was a photographer. That's what structural biologists are photographers. You spend, if someone runs a Western blot or someone determines a structure, theoretically that's the same thing. You run one assay, right? That's it. But a Western blot can be done within a day. A structure might take you 5 years and you're already there. And so, particularly as a structural biologist, you're so focused on the methodology and optimizing the methodology, making this protein better, testing different constructs, that what you're completely losing sight of and what you've never really been taught is hypothesis-driven research, which I guess makes up, like 90% of research. And as these tools become more and more automated, as, now cryo-EM, is basically moved into facilities and more time can be spent on the actual biology and coming up with hypotheses, this one and done structural biology is going to go away if it hasn't already. And it'll, it reverts more and more back to what it's supposed to be. And that's about asking and answering very interesting biological questions. And I think in a structural biology lab where you've spent your career on just optimizing something, that isn't for everyone. If you're a methodology developer, sure. I mean, this is very important, and there's always going to be more to be done. But that wasn't for me. I want to learn more about the biology, the pharmacology of GPCRs, and so I needed something else. I couldn't just learn more about taking pictures.
I like the way you put it, and I also I see molecular pharmacology and structural biology, the two sides of the same coin, which get us closer to understanding how a GPCR functions and what it does, and translating that information into a disease state.
Absolutely.
I also love the fact that you very specifically knew that you wanted to go the PI route, the academic route, and then you picked a postdoc that would allow you to acquire more knowledge, learn new things. And to your point, I think, at least for me, doing the postdoc was an important personal growth step. And also as a scientist, I think I did a master's in Montreal, a PhD in Montreal. In the master's, during my master's, you kind of learn how to pipette, not kill your cells. And towards the end of my PhD, I actually learned to think scientifically and be like, hey, why don't we try something new? And what if? And then the postdoc, at least in Tom's lab, was challenging, best 5 years ever, because I did bring in a set of methodologies and applied them to questions that were not asked in the lab. But at the same time, when I left, a lot of these methods are still being used in the lab. And starting from zero in Tom's lab when it came to all of these signaling assays, it was hard. It was really hard. But at the same time, it was so much fun at the end.
I mean, we're all here for a challenge at the end of the day, right? I mean, very few people go into a lab and they work on something that's basically already been figured out. I mean, how many times do you go to a conference and someone starts, their talk with, we're just beginning to understand. I mean, no one works on something that's already completely known. We're here to figuring things out. We're solving puzzles. That's really what it is. And I mean, you can work on GPCRs all your life, and you'll never contribute really to a big drug or something like that. But it's about trying to solve a puzzle, because you don't know at the end of the day what you're going to discover and how that applies to the grand scheme of things. And I mean, a lot of discoveries that are now like major treatments or that have the greatest promise are like serendipitous discoveries. CRISPR, I mean, it was studying the immune system of I guess it was a guy at Danone who was also credited with that, studying the immune system of lactobacillus, for instance. Or I mean, if you go back to Röntgen, the guy who discovered X-rays, I mean, it wasn't the intention to find radiation and look into people's bodies if there's something wrong with their bones or something like that. It was just the dude staring into the sky and trying to figure out what kind of radiation hits us. And then we've, over the years, developed this into, fantastic medical equipment. So if you're not in it for the challenge, you're not in it for solving puzzles, the idea that you're going to, save lives, I think that's not the concept of a scientist. That's, I mean, you want to go the MD route. You don't know what you're going to find. It might mean nothing. It might mean a lot. In most cases, it means a little bit. And the grand scheme of things, your little bit is going to contribute.
Exactly. So after your time in Bryan's lab, you knew that you wanted to become a PI, but how did you plan for it? Did you think about going back to Europe? How did you end up where you are today?
I did. I did think about going back to Europe, but at the end, things always turn out differently. It wasn't really super high on my list, but I considered it for some time. But I also got a little bit scared by the fact that I now spent about 8 years of my career in the US, and I at least fooled myself in thinking that I would know the system a little bit better than I know it in Europe. That's not entirely untrue, but as a postdoc, really working hardcore on things and maybe submitting a fellowship here and there, who knows anything about like grant writing until you really do it as sort of kind of a full-time job. The sad truth of the matter is that faculty jobs are very rare, and there's very few of them, and they're hard to come by. And so that isn't as much of a, hey, where do you want to go? What do you want to do? As it is, what is available to you? And the options are rather limited. So in many cases, it really needs to be a very good fit. And one of the first attachments I had, or the first contacts I had with Mount Sinai was Marta Filizzola, who is my colleague here at Mount Sinai. You had her on our podcast before. And I met her for the first time at Brian Kabilka's meeting out in Hawaii. And I remember talking to people from a company there, and they were like, hey, I mean, once you're ready, I mean, would you be interested in working for us? And Marta immediately intervened. It's like, no. You don't want to do it. You want to come and work for us. And I'm like, that kind of stuck in my head for the next few years. And so once it came time for me to look around, Mount Sinai was one of the early places I looked at. And another colleague of mine, John Jen, he had just transitioned, I think, a year and a half or so before me from the University of North Carolina to Mount Sinai. He runs a big chemistry group, and I didn't interact with him a lot, but I did interact with him when I was at UNC, and we published together. And so there was a certain level of comfort of knowing people like Marta, of knowing people like John Jen. John Jen having done the same transition, albeit on a very different level. So Mount Sinai was very high on my list at the time, even though there were a number of other places that I was fortunate enough to get offers from. And so yeah, that's kind of everything else is history. Here I am.
That's awesome. All right, back to the science and back to GPCRs. Yeah. And you've seen my questions, and I asked this from everyone. Do you have a favorite GPCR?
Do you have a favorite child? I mean,
that's no, no, that's hard.
It's a difficult question to answer. If I've only ever worked on one, I could probably immediately point to it and say that. But that's a very, very difficult one, to answer. In my mind, the short answer would be no. But I could give you a number of them and tell you why I think they've had a specific influence. If that's okay yes. with you, I think
absolutely.
I think arguably, I think very, very few people will dispute the fact that the beta-2 adrenergic receptor and rhodopsin probably have done more for the field of GPCRs than all the other receptor work combined, certainly in the structural sphere. Completely different story when it comes to molecular pharmacology, biology, pathophysiology, and so forth. It's not like rhodopsin is, has any more diseases associated with it than any other GPCR. That being said, beta-2 was also my first. It was the first structure I'd ever solved, and I'm still very proud of that paper because it kind of showcases how lucky you need to be, because it was a year into my Ph.D. that I published a JAX paper with three beta-2 adrenergic receptor structures, and in retrospect, I had no clue what I was doing. I was solving structures, okay, fine, but it just feels like a haze, and I don't know, there's just something to that time, to that phase, that makes me think very fondly of that receptor and what we learned. But obviously, to make the bigger case, I think, like I said, beta-2 just taught us a whole lot about pretty much everything on the mechanistic side of GPCR structure function. If I were to talk about, actual learning about psychedelics, which is something that I've worked on for quite some time, it would be the 5-HT2B serotonin receptor, which ironically isn't even expressed in the brain. So how the hell does that have anything to do with psychedelics? But it was the first big serotonin receptor that was solved. I don't know why I'm saying big, but it was the first receptor, a serotonin receptor that was solved, and I stuck with it, and it helped us, then later solve a structure bound to LSD, which was the primary psychedelic I was working with at the time. And so I would probably point to those two for the two different reasons, being my two favorite receptors, if that's a number that's acceptable.
It is absolutely acceptable. It's funny because when I started the podcast, I was talking to Silvio Gutkin, and I did ask him the same question, and he said, "Well, actually, it's not a GPCR, it's G proteins." But he said, I cannot remember if he has 2 or 3 kids. I think even daughters, if I remember correctly. And he said, I cannot pick a favorite. So we went through.
I didn't steal that. I did not hear that part. Okay, I'm glad I'm not the only one.
No, you're not the only one. And we went through all of these. And for example, Michel Bouvier's answer was that he does not have a favorite. Is more interested about the interest is around the question, and the receptor is just a modality to answer a set of questions.
That's a very diplomatic answer there.
Yes. That's
sounds about right.
But exactly. But you've seen his work. He's done, research on so many different receptors. There's no specific track. I think the common theme is GPCRs.
Yeah.
And now, what are you working on right now? Since you mentioned the beta-2 adrenergic, the serotonin receptor, did you continue working on those receptors, or where is your research going right now?
I am still working. I'm still working on a number of GPCRs with a particular focus on serotonin receptors, and we've started transitioning a little bit more into trying to figure out which drugs actually have unexpected effects through serotonin receptors that have previously not been known, and we've been making some decent progress on that over the years. Which we'll hope to turn into some publications soon. So I've kind of continued along these lines, but I've taken basically a big page from the book of the Bryan Roth world and looking more from the drug side than from the actual receptor side. And when you do that, then all of a sudden it becomes clear to you that and this isn't blasphemy here, that GPCRs aren't the end all be all, and we've been looking at drugs now from a much broader scale. Obviously, if you consider why GPCRs are such amazing drug targets, one thing of many, and I don't want to go into a 2-hour rant here. One thing of many is that a lot of drugs are similar in size, similar in chemical nature than the actual hormone, the actual endogenous ligand, like in the case of serotonin, certainly the neurotransmitter serotonin. And when you think of that, there's a number of other proteins in your body that obviously touch that neurotransmitter. I mean, it's enzymes that need to make it. It's enzymes that degrade it. It needs to be transported from A to B. It needs to be shuttled around. And then you start thinking, hey, what about these serotonergic drugs? Do they just hit these receptors? I know that's how they're supposed to work, but do they also hit all these other proteins? And do they hit the transporters and the enzymes? And that's kind of where we've gone a little bit, like a little bit of not away from GPCRs in that sense, but broadening our basis. But we're still very deeply rooted in GPCR work, and half my lab still works on GPCRs, and it's predominantly serotonin receptors at this point we're working on. But there's 13 of them, so we have our hands full with that for sure.
That's amazing. I love the approach, and you make a great point about, yes, we talk about GPCRs, we talk about drug or ligand GPCR interaction, but these are not made in space, and they come to interact with only the GPCR in the human body or in an animal model. These need to be produced, transported, modified, activated in order to get to their final destination. And I think we've been focused, or at least I have been very much focused on what happens once the interaction happens, and then what happens in the cell. But the before moments are also very important to.
I mean, let's not pay too little attention to that. I mean, that is how drugs work, right? I mean, we know for a fact that an opioid receptor binds to opioids, gets activated, and that causes analgesia, just like it causes respiratory depression and it causes the death of way too many people. But a beta blocker blocks beta-2 and beta-1 adrenergic receptors, and that's how it acts. Like, that is a key interaction, but it does not explain a lot of the side effects. And that's kind of sort of the way that we're going a little bit, that you have more of a holistic, approach and get a more, polypharmacological view on what these drugs do. And that's again rooted in the fact that I've come from GPCRs, I've gone into drugs, and now I'm looking more from the drug side rather than from the GPCR side.
Which is absolutely a valid point as well, because you could be making discoveries as to how to make better drugs that are more specific, that stabilize a specific conformation or activate a specific subset of receptors by knowing what happens to that drug before it actually reaches the receptor.
Exactly. That's exactly right. Yeah.
So you did mention GPCRs as good as drug targets, and I guess the answer is they're still good drug targets.
Yeah.
Any comments around that?
I mean, not really. Like the track record stands for itself. We're always facing this criticism that, GPCRs at the end of the day, while they make up a lot of the drug, basically the realm of the drug targets, Where's all these new drugs for GPCRs? And then, I mean, I can tell you, I'm getting bombarded with them by just watching TV. Every single time there's like some, you know, some ad coming up with some drug I've never heard of before. I Google it, and the chances are, 40% of them tend to be GPCR drugs, and there's still new ones. So make no mistake, GPCRs are fantastic drug targets. I think one of the things, and again, I think that's something that's changing, and that's changing for better, is that obviously, as we're understanding more and more about these GPCRs, we're starting to look at the more understudied ones. So previously, it was all the big famous ones. I remember when I started, in Ray Stevens' lab, it was all about, the serotonin, dopamine, opioids, cannabinoids, and then glucagon, like the B class came in. But there's this entire dark side of the moon, so to speak, of, like completely neglected proteins that are, through some knockout studies, maybe there's some implications of physiology, but some of them are not even conserved between, humans and rodents. So what do you do then? And I think this is a little bit where the field is going and needs to go, is to really illuminate these understudied GPCRs, because, as great as drug targets they are, if you actually run the numbers, like a majority of the drugs, you could probably use the power law there, or Pareto's distribution, like 80% of the drugs only target 20% of the receptors. And there's plenty of reviews on this. So while they're biophysically perfect for drugging, what are the chances the other 80% of the GPCR universe aren't also great drug targets for other stuff? I think it might have a little bit to do with the fact that it's somewhat difficult to get grants on some of these, but there are some mechanisms, illuminating the druggable genome and so forth. But I think as far as drug targeting goes, I think that's where all the big potential is, and like understudied GPCRs, because they are perfect for being drugged with small molecules, which still is the primary choice of drug for people.
You made a couple of great points, and I have to echo what you said about Googling drugs that you see in commercials. And I think the most prevalent one is the GLP-1 peptide, the semaglutide, and all of its forms for type 2 diabetes. I also think that, GPCRs are good drug targets, and you mentioned, getting grants and getting the funding. I think that that's really one factor that doesn't allow us to spend enough time on other targets than the ones that we've studied before. That being said, I think there are more tools. We were doing, cryo-EM definitely changed the face of getting structures, the way we get structures. Last week with the adhesion GPCR papers that came out, there was not one, but 4 papers that were published. I think it was two receptors. And I think it's just amazing. We have more and more tools. Measuring the pharmacology is getting up to speed as well. I remember doing cyclic AMP measurements with radioactive, with tritium, and you had to purify the cyclic AMP on columns and sets of columns, and now you do it in 384 well plates, and like that.
Fluorescent layer. Yeah.
Exactly.
Yeah, no, it's, technology drives discovery. There's like no doubt about it. The initial GPCR structural revolution was all driven by LCP crystallography and the fusion technology, and then now cryo-EM has ushered in a second, phase of that. Certainly technology is always going to be key, and who knows what's around the bend. Right, in the next 5 or 10 years, how that's going to revolutionize GPCR research, but also, other research.
Is there any advance or any technology or technique that you wish, you had a magic wand and you would create it to help, advance your work or advance the field?
That's a very fair question. Obviously, being a structural biologist, I'm very biased on that. At the end of the day, I think people like pictures. They do. Chemists like pictures because it really helps them, make molecules to just boil it down to the simple essence. So from a theoretical point of view, we already have some of these tools. I just wish they were a little better. As much as I like cryo-EM, as much as I like X-ray crystallography, they both have their severe disadvantages, and it still requires a lot of biochemistry, a lot of manipulation, requires a lot of like excellent sample preparation and all that. So if I were to design something, take the Elon Musk approach, I wish at one point there was an app in my in my smartphone, and I could just take a picture of my vial, and I would know exactly what the GPCR looks like in that very state, and I could divvy it up in the distinct different states that it would adopt in the way it's sitting in solution or in a membrane. But obviously the holy grail would be some form of structural biology within the native context at single GPCR resolution, with high enough resolution to really resolve atomic details. So that's kind of the technology that I obviously would love to see. And I think there's technologies out there like free electron laser, well, not crystallography, but they're obviously working on making it single molecule applicable. But who knows how long that's going to take to get to that point. But if you talk about, well, like you said, what you did 10 years ago, radioactive CMP and all of that, you always think, okay, you'll never see that day, and then 5 years later, you're like, I can't even believe I did it any differently. Yeah. But that's life at the end of the day.
Yep.
Right? It's the same with everything. I remember the pre-internet days. That's how old I feel. So.
Yes, it's funny because I do too. And when I think about, for example, the first iPhone, and it seems so easy right now because you can do so many things from your phone, but 10, 15 years ago, it wasn't the case.
Yeah.
So
it's very difficult to predict what's going to happen in 10 years. You watch whatever Back to the Future movies from the 80s, and they predicted, like hover skateboards, but it didn't predict the internet. So it's impossible,
Yeah.
And then there's people working on God knows what. It all comes back to you just got to, as you're solving puzzles all day long, you got to look everywhere because you never know what you're going to find.
Agreed. I love the the Back to the Future. Every time, it's funny because every time I watch it, or I watch any movie that talks about the future, and they pushed it to 2022 or 2030 something, and you're like, nope, we're not there yet, and we're not going to be there in 10 years either. So no hoverboards yet.
Nope.
All right, super. So we talked a little bit about advice for junior scientists on how to pick their own lab, to do for their training, or going the PI, the academic route, or the industry route. Any other advice that you would have for young scientists if they wanted to contribute to the field?
Pursue your passion, right? This is a tough business, and I'm not talking about people. I'm talking about the fact that you're going to fail. You're going to fail all the time. There's statistics and there's numbers on that, and you can look them up for yourself. But most of your experiments are going to fail. So the only thing that's going to keep you glued to what you're doing is that tomorrow morning you're going to get up and you find it in yourself to go back until you have that one beautiful day where you have that piece of data that you've always wanted, and it is going to happen. But that being said, that comes in many shapes and forms. Don't be too narrow-minded on, academic inquiry is literally the only way to do it. There's great research being done in, startups. There's great research being done even, in major pharmaceutical companies. You have the COVID vaccine within, like a year's time, and that's not due to a single person. That's because people came up with it in an academic setting, and then people managed to adapt it to something that was applicable in the real world. And here we are, and most of us, at least the people that can listen to the stream, certainly survived. So, just explore your options, but do something that you're passionate about, that you like, and don't be deterred by failure, because that's going to happen, and it's going to happen to everyone. It's about getting up the next morning and just still being interested and being motivated to do that. GPCRs, they're fantastic, but they're also not the end all be all. And if you think that there's any given field in the world that, is at the end of the rope and it's all been solved and it's all been done. You don't know science. There's always more to be done. We're very serious about our job security. So.
Agreed. I love that. And I think you make a great point about failure. Most of the experiments that you're going to do are going to fail. And I think in some way, we're all a little bit, all of us are, I'm looking for the right term. Not adrenaline, but what's the hormone that makes you feel good? Dopamine.
Dopamine.
Is it? Yes. We're all
or serotonin.
Junkies.
Both
Yes,
of them.
we're all
We'll do it in
junkies.
different ways. Yeah, junkies.
Right?
Yeah, that's a dopamine.
We're all junkies for
right
that.
there. Yeah.
For that feeling, for that I did it. It finally worked. And I think if you can get up every morning knowing that yesterday it didn't work and the week before it didn't work, and think to yourself, okay, maybe today I'm going to go back and see what I can do better. And it's incremental, and sometimes you don't know. It's just a matter of, luck, but also a lot of hard work.
Oh yeah, nature is a beast. You better come armed and prepared. It's tough to get these secrets and to discover things. Yeah.
Agreed. We also talked a little bit about the receptors that you like, and you can't really put your finger on which one is your favorite, but totally, I think it's a normal and it's a great answer. During your time in the lab, whether it was at your master's, Ph.D, postdoc, were there any moments where you discovered something or something happened that changed your perspective? I'm thinking aha moments here. It doesn't have to be scientific either.
I think one of the most rewarding things so far has been mentorship. I just have to point that out. And I think many people will mention that. I don't know how sincere people are about this, because obviously science is somewhat of an egocentric thing, right? Like you're really buckling down and you're trying to figure out something, and it really sucks if someone else figures it out first. Because at the end of the day, someone's going to figure out the problem that you're working on. It might be you, it might be someone else, but someone will. So, the idea of being unique, it's a tough one to deal with. But the one thing that I found more rewarding because of that, because of the, that to some extent somewhat irreplaceable, because a lot of people just work on these things, is the mentorship aspect. And the first time you see someone who you've been training for, weeks, months, years, it really depends, to make a discovery. And I've had some very good fortune here with some of my people in my own lab that basically, without me telling them anything, like picked up their own projects and were very, resourceful about things. I've had students buy really interesting, like supplements off of Amazon and discovered amazing activities at GPCRs we didn't know before. And the fact that I didn't have to tell them to do that, but that they just went ahead and did it because they read something somewhere and then came in and showed me this graph of data. It was cool to find that, but it was even cooler to be like, okay, this person just did that because of, like something that I taught them, or it really helped them to get to that point. And I don't know, that was kind of an aha moment where for the first time I was like, okay, maybe there's more to just sitting there in the corner yourself and playing with your little puzzle. It's mentorship is a key aspect of all of this. And in an academic environment, there's always going to be mentorship because it's not technically a production facility, right? We're all educating PhD students, we're educating master students, interns. I've had master students, I have high school students, college students, That was something eye-opening for me that goes beyond science, that I've really never known how much I enjoyed mentorship. It's so rewarding.
That's amazing. And I really do like the way you think about it and you approach it. Because at the end of the day, even you mentioned this project and the student, you gave the space to experiment and do something out of the box. And that's amazing.
Yeah. Sure. I have to apologize. The New York sun is strong. I don't know what's going on here right now.
Well, that's okay. I actually had to pull the drapes in because because it was in my face as well. But yeah, at least it's sunny. We cannot complain. It's
Yeah.
not
Oh,
San
no,
Diego,
I'm not
but
complaining.
it's sunny.
Not quite. It's pretty good. That's pretty good.
We'll take every ray of sunshine. You were talking about your lab and about the people in your lab. If and when you do have openings in your team, where can people find you?
I think typically the new game is now social media, so I will most likely make an announcement there. If not, you can find my lab obviously on Mount Sinai and Mount Sinai's webpage, but I have my own lab webpage. I'm usually up to date with the postings there, so that's wackerlab.com. And you can find openings there. I currently don't have any, so the timing isn't too great on that. But there's certainly something to be announced on there once time comes around.
Perfect. Well, thank you so much, Daniel. I really enjoyed our chat.
Yeah, likewise. Thanks for having me. That was great.
Thank you. Take care.
Thank you. You too. Bye-bye.
Bye.
Thank you for joining us and listening to this podcast episode. We'd like to thank our guest, as well as our team members, Attila Forrest and Ines Pinheiro. Please make sure that you subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, please leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Don't forget to check out and register for the Dr. GPCR ecosystem at drgpcr.com/ecosystem. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.